Hello, everyone, and welcome to a comprehensive study on adeno-associated virus analysis using a 3-micron monodisperse size exclusion chromatography column. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, please type them into the Ask a Question box and click Send. We'll answer as many questions as we have time at the end of the presentation, and you can submit any technical issues there as well if you are having any trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Kav Ma, Staff Scientist at Thermo Fisher Scientific, and Ashleigh Koontz, Global Product Manager at Thermo Fisher Scientific. For a complete biography on our speakers, click the biography window on the left side of your screen. Ashleigh, you can now begin your presentation.
Thank you. Hello, everyone. Ashleigh Koontz, and today we will be going over a comprehensive study of adeno-associated virus analysis using our newest 3-micron monodisperse size exclusion chromatography column. I'm the Global Product Manager for BioLC columns at Thermo Scientific, which includes DNA Pack, ProPack, MadPack, and now our new SurePack family. I'm here today with Kav Ma and looking forward to going through this presentation with you. So the analysis of AAVs, adeno-associated viruses, are a very fast-growing market in gene therapy, and we're looking for safe delivery of genetic materials into target cells. That effectiveness is going to rely heavily on purity and homogeneity of the AAV stereotype and that vector population. Aggregation plays a significant role in production and quality control, and we can focus in on size exclusion chromatography as a preferred method for detecting and quantifying AAV aggregation. The standard size for AAVs is 25 nanometers, and that is going to take strong consideration into how we designed in and developed our newest suite of size exclusion chromatography columns. The new SurePack Bio 550 SEC MDI columns were developed for you as biopharmaceutical scientists in cell and gene therapy to help quantify and identify aggregation and monomers in your methods for analytical development and QC. This new wide pore SEC column is going to provide exceptional resolution of monomers and aggregates. And with these new 3-micron monodisperse particle, it's going to enhance that resolution and reproducibility. And all of that media is packed into a hydrophilic inert hardware. This product was launched this year in July. And as you can see, we have a variety of different formats to support your analytical needs. So this new product family is manufactured in our campus in Vilnius, Lithuania. It houses MDI technology, which is monodisperse inert. This is combining our advanced monodisperse particle platforms inside of bioinert hardware. Our Lithuania manufacturing team has wonderful capabilities from LC processing and distribution and has wonderful ISO certifications, and also manufactures our newest ProPack 3R columns and the new SurePack Bio SEC columns. For our new SEC columns, we do offer the most advanced technology for size exclusion chromatography on the market today. We're excited to offer this within the silica portfolio. So we have a monodisperse 3-micron particle size with that wide pore size of 550 and that bioinert coated hydrophilic stainless steel hardware. The difference here is those monodisperse particles, which are going to provide a ton of separation benefits in your resolution and efficiency. And we want to target the larger modalities in biopharmaceuticals. So we're seeing AAVs as a good target market for these, but other biomolecules as well. So if you're looking at that range between 300 to 8,000 kilodaltons, this should be the separation, the SEC separation for you. We will not be discontinuing any other of our SEC products, But as you can see here, we do have others that are in various pore sizes. But again, if you're running AAVs or large biomolecules, look to that SurePak SEC column. We have a variety of resources that we can offer you, including our application note, which will go into further details, as well as quick start guides and product manuals, and a flyer to kind of round out the offerings that we have in other chemistries to support you with AAV analysis. And now we're going to dive into our technical background, and I'm going to pass it over to our lead R&D scientist on the project, Ka.
Thank you, Ashley, for the nice introduction, and thank you for covering the first part of the presentation. My name is Ka, and I work in Thermo Fisher as a staff scientist who focus on application development for all of our bio-LC columns. So I am going to walk through this for the technical background of the new SurePak Bio SEC MDI column. As Ashley has introduced this column, one of the major applications is going to be for EV therapeutics. So most of the audience here should be pretty familiar that SEC separates the molecules based on their hydrodynamic radius. If we look at the picture on the right side, we can see if we have a group of samples, the large molecule gonna elute first because most likely it's gonna only travel through the interparticle space. With a smaller particle, it's gonna also travel within the inner particle space, which has longer retention time. And for AAV therapeutic, it is very important that we understand the aggregation if the AAV is pure enough. We have the monomers and aggregation titer. And this is a very essential part for the CQA from both R&D AD to commercialization and in manufacturing and QC. So the aggregation can be caused first by manufacturing related reasons. So for example, like in the expression system, growth conditions, purification, formulation, and also it can be caused by stress related reasons like heat, freeze and thaw, cross-linking, protein concentration. So, and the aggregation can result in incorrect drug dosage and decrease the solubility, and also result in activity loss and undesired immune response. So by employing the SEC method, this can greatly help us figure out the aggregation of EV samples. So this is just a brief introduction of the SurePak column, the physical properties. So this new column uses a 3 micron silica particles as the stationary phase, and the particle is modified with diol group, so to prevent secondary interactions. The average pore size is around 550 angstroms, which is about 2 to 3 times of the AAV particle size. We offer 150 millimeter for the column length with 3 diameter, 2.1, 4.6, and also 7.8 millimeters. And all of these formats will have the guard, which is 30 millimeter length. And as Ashley introduced, the column is housed in a stainless steel hardware coated with hydrophilic inner layer, which is our latest inner technology. Typically, we run this column between 20 to 60 Celsius temperature, and the maximum pressure will be 3,500 psi. Be handled by most of the modern HPLC systems. PH range is 2 to 8. On the right side, the bottom pictures are a real SEM scan of the new particles. As you can see, the left is the monodisperse technology, which we have precise control of the particle size. The white bar is the 8 micron legend, so you can see the particle size is precisely around 3 micron. And on the right side is more conventional polydispersed particles, which you will have the tiny fines and also larger particles. So both of them are gonna give you unwanted chromatogram results. So for example, the little fines might cause higher pressure, and the large particles might reduce your resolution or efficiency. So the recommended operating conditions for the SEC separation to couple with this new column, we recommend a fully biocompatible Thermo Fisher Vanquish system. And for the different formats of this column, so for the 2.1, 6, 7.8, do I have a different maximum flow based on the ID? In order to get the optimal separation results, we also recommend the user to pair the tubing ID with the column as well. So if you have a thin column like 2.1 millimeter, we recommend you 75 micron wiper so that you can minimize the extra system volume. And also we have the nano wiper and the nano wiper finger tight fitting part number listed here. So people, if you're interested, you can feel free to order. And we'll also show some data to help prove and show that the extra tubing volume actually matters a lot in your separation. So this is an example of the separation using the 2.1 times 150 millimeter column. One is using an inlet and outlet capillaries nano wiper of 75 micron. Bottom one, the blue trace, use 100 micron tubing. Here, for the radical plates, is not optimized using a wider tubing. We can see there is about 10% efficiency loss compared to using the optimized tubing ID. Also for the peak width for albumin, which is this peak number 4, you can see the peak on the black trace is significantly sharper than the bottom one, and also we have better resolution here for the albumin aggregates, the peak number 3. And if we look at the data here, we actually have the peak sharper by 30% just by replacing ID tubing to a thinner one. And also for efficiency-wise, if we look at if we look at the cytoglobulin peak width, again we have 18.6 seconds, which is the blue trace, versus 12 seconds with the black trace. So the peak width is about almost 40% sharper. By choosing the proper tubing ID, we can help improve the resolution of the monomer and the aggregate separation. We briefly discussed before, the new product use 3 micron monodisperse silica particles. By employing the monodisperse technology, we will gain precise control during media synthesis, also column packing. This will also help greatly increase the column-to-column and the lot-to-lot reproducibility. And all the particles are covalently modified with hydrophilic surface to prevent ion exchange sites. And the particle size, the pore size is 550 angstrom, which is 2 to 3 times of the size of AEV. This is the rule of thumbs when we choose the pore size for SEC analysis. You want your pore size to be about 2 to 3 times of your. We also introduced before, this new technology media is housed in a hydrophilic stainless steel coated inert stainless steel hardware. So here I want to focus more about the method development and also the reason why we choose 550 for this product for AEV application. When we were doing at the screening stage, we tried a couple candidates with different pore size. So we have a 300 angstrom here, we have 450, we have 550, and we also have a 1000 angstrom candidate here. And we can see when we're separating AV5 in this case, when the pore size is about 300 angstrom, we cannot separate the high molecular weight species peak, which is the aggregate here. We only have one main peak for the AEV monomer. Once the particle pore size is large enough to 450, We can see here we have the high molecular weight species peak separated, and the resolution between the monomer and aggregates here is 1.47. And for the 550 angstrom, we have the resolution increased to 2.34. Once we are at the 1000 angstrom pore size, we can see although we still have certain separation for the aggregates with the monomer, it's not baseline separated anymore, which indicates the pore size is too large for this case. So based on these results, 450 and 550 will be very promising candidates for the AAV application. So we did more comparison with other samples with these two different pore size. And we can see here on the top is the SurePak MDI SEC column, and on the bottom is a 2.5 micron polydispersed SEC column with the 450 angstrom from another vendor. And if we look at the three samples here, AAV3, 5, and 8. So first, all of them will have a better separation for resolution-wise between the monomer and aggregates. For example, AAV3 have 2.63 versus 2.1, AAV5 had 2.4 versus 2.1, and AAV8 is 2.5 versus 2.2. And also we obtain sharper peak, which is a number here, peak with half height, 167 to 1 versus 193, and it's also the rest of samples. We actually obtained sharper peak thanks to the monodispersed particle, and also we have better resolution with the 550 pore size. And another thing which is also very important is the detection method. So here is a brief comparison of using UV and the fluorescence detection. We also will cover some of the SECMOS results in later of the deck. But if we just quickly look at this, we can see, so each detection method will have its own benefits for sure. On the top trace, this one is using UV detection, and blue trace is 260 angstrom, 260 nanometer. The black trace is 280, and because all the sample we tested here are full sample, that means the AAV will contain the DNA inside. And the DNA actually have stronger absorbance at 260. So we can use this feature to make sure the identity of the peaks. Look at the zoom in here. So this aggregates peak, we have stronger 260 absorbance. The monomer peak, we also have stronger 260 absorbance. And introduce the large sample where you lose first. So this one will be the aggregates, and this one is the monomer. And we also have a third impurity peak, which is eluting after the monomer peak. This peak also have a stronger 260 absorbance, but it doesn't have any fluorescence signal. So our assumption is this one is extracellular DNA, which travels all the pores and inner and interparticle side pores. That's why it elutes actually later than AAV monomer. So if we use UV detection, this can help us with peak identity. And if we also we can also use fluorescence as detection method. So one benefit is fluorescence will have much higher sensitivity. So this will help you to consume much less sample. So we can see for UV, we injected 1 microliter, and for fluorescence, we injected 400 nanoliter, which is half. So once you have the peak identity confirmed, you can definitely feel free to switch to fluorescence. So here is some additional examples. So we have AAV3, AAV5, AAV8, and AAV9 here. The bottom is the enlarged chromatograms of the aggregates peak or the impurity peaks, so that we can identify if the peak is the AAV peak, and we can see all of those are baseline separated. So the column definitely help with the separation of aggregate and the monomer with different serotypes. Another thing people can choose depending on the user's need is a flow rate for your method. So for example, on the left side, we have a separation with a slower flow rate. And on the right side, we have a separation faster flow rate. So this one I remember is 0.05 mil per minute. This one is 0.3 mil per minute. And we can see when we're running a slow flow rate, we'll have a much higher resolution. 1 versus 2.2 using 0.3 mil per minute. And also we'll have higher sensitivity here, which means you have stronger signal compared to using a fast flow rate. But the method you can see is actually 45 minutes. So if you are focusing more on resolution, you want to get a better separation, we definitely recommend you to use a slower flow rate with a longer method. But sorry, this one is 0.6 mil per minute. If we use a faster flow rate as 0.6 mil per minute here, we can see the separation will be reduced to only 4 minutes instead of 40 minutes. Although the resolution is sacrificed a little bit, 2.23 is still baseline separated. So if you're in a fast-paced environment like a QC lab, you can have flow rate with a very high throughput method. And this one is a table of impact of flow rate on both runtime and resolution. So on the left side is the runtime. So as you can see, from 0.05, the runtime is about almost 30 minutes. Increase the flow rate up to 0.6, and your runtime will only be 4 minutes. And in the meantime, your resolution will also be decreased once the flow rate comes up. So it's very important to choose the flow rate based on the user's need and balance between the runtime and the resolution. This slide, I want to talk about the format and the tubing selection. So this is more data about choosing the right capillary tubing. So here we have a 2.01, 4.6, and also a 7.8 millimeter ID columns. Flow rate and injection volume is scaled based on the from ID. And the relation between these three formats, once the parameters are scaled correctly, the relation is very similar. Is around 0.03. It's 0.0313. This one is 0.367, and the resolution is all around 2. So they're all baseline separated. And here is more data showing that if we use a 100 micron Viper versus a 50 micron Viper, we can see we have a much sharper peak, and the resolution is also greatly improved. It's always very important to optimize the tubing selection for the system and minimize the extra system volume. And this column also is very good with large sample loading. So for example, some application like SEGMOS, you will typically inject a lot of samples. It's definitely the right candidate for it. Here I want to show the sample loading effects of injection volume. So we have 4 trays here. The back trace is we inject 1 microliter of the undiluted AAV8. Blue trace is we inject 10 microliter of a 10-fold diluted sample, and pink trace 20 microliter of 20-fold diluted sample, and brown trace is 40 microliter injection of 40-fold diluted sample. So in this case, the actual amount of AAV injected on the sample is same. The only thing we're changing is the injection volume. And we can see, although the injection volume is 40 times larger than the 1 microliter injection, the peak weight is only increased for about 26%. So this column is definitely a very good, this did also shows a very good capability of the column to handle large injection volume. And we also have very similar peak height for the same sample loading amount, although the injection volume is different. So here I want to show some of the SEGMOS data performed by our colleague in Florida site. So we all know SEGMOS is the gold standard for AAV characterization. This is a crucial technology in product development. as the feature of rapid and accurate. And typically, the SEGMOS requires larger sample volumes than SEC UV and very sensitive to particulate released from the system and column. With the latest hardware technology we employed for this column, we can make sure that there is no particulate release from this column, and thus can help you reduce the equilibration time for your SEGMOS system. And on the left side is the molecular weight data for the AV sample. We have the trimer peak here, we have the dimer peak here, we have the monomer peak here. And on the right side, we can see we also performed this experiment using a column W, which has a 300 millimeter lens, 5 micron ID. And because our column is 150, so the shorter lens can enable us to have a higher throughput method and reduce the overall runtime by more than half. And also we can inject 50% less sample compared to a longer column. Even though our method is faster and we're injecting less sample, we can see the resolution between the monomer and the dimer is 1.72 versus the 1.26 of the long column. So all of the experiments data here are operated by the Vanquish Flex Horizon LC system, coupled with the MOS detector. And the SurePack column enables smaller sample volume and also gives us a higher throughput. This page, I want to also talk about the lot-to-lot reproducibility, which is a very important feature most of the users cares about. So here we have 3 different columns with 3 different lots of the raw media, and do the chemistry modification visually and packing them into the column. And we can see for the AV3 sample analyzed using the 3 different lots, we have a retention time with a 1.9% of deviation. We have a resolution around 3% deviation, and they're all around 2.6, 2.45, and 2.5, so baseline separated. We have the monomer relative peak area about 93 to 94% of 0.3% deviation, and the aggregate peak around 6% with 5.7% deviation. So this SurePack bio monodisperse column definitely enables, and we have consistent results for each column and for the different lots. Next section, I want to also briefly talk about comparison with columns from other vendors. First is a recap of the SEGMOS data we just discussed. So the column W we used for the SEGMOS for the comparison is 5 micron versus ours is 3 micron. The pore size is very similar, 500 versus 550. The column length is double. So this will require longer runtime, which is 30 minutes versus 12 minutes, and injection volume will also be doubled because of the double. And the results, as we briefly discussed, we actually have better resolution and sharper peak, more than half of the runtime. The SurePack Bio SEC MDI definitely delivers or better resolution of critical pair in half of the runtime. This one is a comparison with column G. Column G is a 450 angstrom column. And if we look at AV3, 5, 8, and 9 data, we can see that we have better resolution with all the samples. So the SurePack Bio SEC MDI column definitely delivers better resolution than the column G. Next one is column P. So column P is 3 micron with a 700 angstrom pore size. The column length or dimension is the same, 4.6 times 150. And if we just pick one to compare, so for example, AV8, this one has a retention time with column P of around 4.5-ish minutes. And if we look at the SurePack data with a 4.5-ish retention time, which is either 0.4 mil per minute or 0.3 mil per minute, we have a resolution around 2.6. This is definitely better than the resolution, which is all smaller than 2.5. So the SurePack column definitely delivers better resolution than column P as well. Overall, in summary, SurePack Bio SEC MDI columns offer a higher resolution and high throughput to accelerate your therapeutic analysis process. A wider pore of 550 angstrom for separating is a perfect candidate for separating monomeric AEVs from the aggregates. MDI technology, the monodispersed particle and the inert hardware will ensure superior reliability. And in the end, the MDI, monodispersed inert technology, is the monodispersed particle is best for efficiency and the column-to-column reproducibility, and the inert column hardware will help to eliminate unwanted actions. Here is the order information for this column. So we have all the formats and also all the guard formats listed here with the catalog number. We also have the website link, the hyperlink here, in case you want to also look at all the collaterals, the quick note, application notes, publications as well. So this will be the end of the talk. And we're very happy to take any questions.
All right. Thank you very much, Ashleigh and Ka, for your very informative presentations. We'll now start the live Q&A portion. So if you do have any questions, feel free to go ahead and put those in the Q&A box, and we'll get to as many of them as we can as we have time for. So let's get started with ones we've received so far. The first one is, what is the advantage of the monodispersed particles?
Okay, thanks, Matt. I think I'll take that question. Hope people can hear me more clearly now. Seems like there's some audio issue with my laptop. So the monodispersed particle, the major advantage is definitely the reproducibility. As we can see in the presentation, we have way better lot-to-lot reproducibility thanks to the monodispersed property, and also increase the reproducibility for our manufacturing as well. And there are also other advantages, for example, like we're gonna have right now with the monodispersed particles, we're actually using a true 3 micron. So there are no or almost no small fines to increase your pressure. There's not bigger particles to decrease your resolution. So those are all the benefits of using monodispersed particles. Thanks.
All right. Can I use the SEC column for the MABS or other proteins?
Thanks, Matt. Good question. I'll take this one. So for MABs or monoclonal antibodies, you're looking at a size of around 150 kilodaltons, so just less than 12 nanometers. When we think of AAVs, that around 25 nanometers, those modalities are much smaller. So we would recommend a 300 angstrom for something along the size of a MAB. So look to our MAB pack SEC1 column, which is a 300 angstrom pore size. For other proteins, this is going to be very modality specific on size. So when you look at, we have had recent success with a 500 kilodalton recombinant protein vaccine. Really determine what the molecular weight range is for your protein. And if it's in that 300 to 8,000 kilodaltons, the SurePack SEC is going to be a perfect fit for you and your size exclusion chromatography separations. But if it's going to be smaller than that, then look to some of the other columns that we have within our SEC portfolio.
All right, thank you. What does inertness mean?
Yeah, I'll take that one too, Matt. So when it comes to liquid chromatography, inertness is going to refer to the lack of absorption of the analyte onto those wetted surfaces of the instrument flow path. So as column specialists, we understand that the largest surface area volume in that flow path of HPLC is the column itself. So while stainless steel is a robust HPLC hardware, it can be reactive and cause certain sticking or peak tailing or loss of analytes for your recovery. So we recognized that and really wanted to ensure that we offered a very inert solution for you with the column. So when it comes to, and again, inertness being important for several reasons, but biomolecules especially have a potential, if you're in the oligos or other biomolecules like nucleic acids, are known to be sensitive to metals. And so this is going to allow that sensitivity to be eliminated and also provide some additional corrosion resistance. So a lot of these methods are often high salt concentrations. You can go to certain pH values that can cause some challenges there. So with the hydrophilic inert coated hardware, we can eliminate any of those unwanted interactions and provide additional assurances to our customers for maximum sample recovery.
Thank you. Can the SEC separate empty versus full AAV?
I guess I will take this one. So empty and the full AAV has almost identical particle size. I think the full is with the DNA loaded is slightly denser and slightly larger. But currently, I don't think SEC has the capability to fully or baseline resolve full and empty. You might just have them co-eluting as one peak, or sometimes maybe as a shoulder peak, slightly shorter. So based on SEC only, it's going to be very challenging to separate empty and full. But if you couple with SECMOS or SECMOS with RI detector, I think with certain calculation that can help, or with calibration curve as well, that can help you determine quantitation of empty versus full. But purely on SEC is going to be super challenging. Thanks.
I would just to jump in there too. We do have a great application note on our ProPak 3R SAX for ion exchange, and that can certainly help as a tool for empty versus full too. So couple that with your SEC for aggregate analysis, and you have multiple methods that you can help determine critical quality attributes for those various AAV stereotypes.
Yeah, thanks, Ashley.
All right. What is the peak eluting after AAV main peak, and do you know the peak identity?
Yes, I think I'll take this one as well. So that peak, as we can see, so we did both UV and fluorescence detection. So first, this peak eludes after the last AV main peak. So we think it might be something small or stay in the column super long. So our first assumption is it might be extracellular DNA as impurities in the sample matrix. So, and also we can see the peak has higher 260 absorbance compared to 280 nanometers and has absolutely no fluorescent absorbance. So our assumption is that's extracellular DNA, although we actually never did any further other testing to confirm the peak identity since the focus is on AAV. But I think with more orthogonal tests, we can definitely determine what that is. Thanks.
Excellent. So we've got time for one or two more. So if anybody has any other questions in the audience, feel free to send those in. So next or last, depending if we get a couple more in here, do you see less sensitivity with SEC mols versus SEC UV?
I think I will take this one as well. So I would probably not say less sensitivity, but SECMOS typically does require larger sample injection volume. I think based on the detection method itself and the comparison of the new SurePak column with also the column from a very popular actually column option from vendor W, we see no sensitivity difference or worse sensitivity at all. And I think we, as our Florida colleagues performed the test, I think we even have a shorter mass equilibration time before the testings. So, but typically, yeah, if you do stack mass, you will have much larger injection volume for sure. Matt, I think
you're on mute.
Yeah.
I apologize. I would like to thank Ashleigh and Ka. Do either of you have any final comments you'd like to give to the audience?
Yeah, certainly. One, we just want to thank you for your time and attendance today. We certainly hope that we brought you some valuable application results and that will help you in bringing your drug products to market faster. If you have any questions or interest in this column, please reach out to your local chromatography specialist at Thermo Scientific. And we would love to get some feedback from you if you evaluate this column and it's working well for your applications. We're always here to support from a technical method development standpoint, or even just if you have other questions within other LC column areas that we can support you at Thermo Scientific. So thank you so much, Matt. Appreciate it. And thank you, Ka. Wonderful job.
Yes, thank you both. And hope everyone have a good day.
Thank you, Ka and Ashleigh, for your time today and your research. We'd like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, we'd like to thank the audience for joining us and their questions. Anything that we didn't have time to get to today, or if any questions are submitted when it is on demand, those will be addressed by the speakers via the contact information you provided when you registered. If you'd like to request any additional information, please be sure to fill out the survey on the left and someone can reach out to you shortly. This webcast will be able to be viewed on demand. LabRoots will alert you when it is available for replay. And we do encourage you to share that email with your colleagues who may have missed today's live event. And until next time, thank you very much and goodbye.
Yeah, thank you. Bye-bye.